Hi, my name is Will Chadwick, and I'm currently a graduate student in the Chan Lab on SF State campus, and we work in something called fission yeast. So this is a single-celled model organism, and it's called a model organism because yeast in humans actually share 30% of our genes. So that means that many of the genes that we see in yeast are actually the same kind of functional genes in humans. And this is interesting because in my lab, we focus on an organelle in the cell called the vacuole, which looks like this. And so these vacuoles are used for storage and protein degradation. That means they break down things we don't need anymore. And in humans, they're the same things, but they're called lysosomes. So we have in all of our cells these little organelles that kind of break down stuff like that, kind of like the liver of the cell, if you've ever gone out drinking. And what happens is these can sometimes get mutated through various different diseases or disorders, and these are generally classified as LSDs, or lysosomal storage disorders. These can affect up to 1 in 5,000 people in the world. And so there's a large class of them, from everything from like Batten disease, which can kill infants, to Tay-Sachs, which I'm sure you guys have heard about before. And so what we do in my lab is we focus on yeast vacuole quantifications. And what that just means is we're measuring the vacuoles in terms of their volume, surface area, shape, and localization. And as you can see here, there's tons of little tiny vacuoles in Pombi in fission yeast. And so what we do is we quantify these, we measure them, and then we do that same thing in mutants and see the differences and how we can kind of look at those diseases and how they affect the yeast, and hopefully end up eventually looking at how those affect humans as well. Yeast are also used in the biopharmaceutical industry to make things like insulin and vaccines, and they're also used in commercial industries to make things like beer and bread. And so understanding how the yeast control the size and the movement of their vacuoles within their own cells can really help us understand more about how these processes work and how we can both fix them when they get messed up, and how we can also control them enough to perhaps improve production of these things or change the way they do various mechanisms in their cell. That's it.